Honors and awarded to her credits are Fellow of National Academy of Medicine Science, Fellow of Royal College of Physicians of Edinburgh, Ramalinga Swami Fellow, National Fellowship from DBT. She has 6 patents on her name and published more than 90 papers in a peer-reviewed international journal and presented more than 10 abstracts on international platform. She received 9 research project funding from various national and international funding agencies, approx round about more than 12 crores rupees. Currently, she is a peer reviewer in various national and international journals. Her area of working is exosome biology and kidney disease diabetic research. She is also a committee member of different national and international institutes. Now, I would like to request Dr. Swasti ma'am to continue this session.
Thank you so much for this very nice introduction, and I think you have said more than what I have done, and thank you so much. Slideshow. So I tried to keep the title in tune with the overall theme of the conference, that is the sustainable approach in translational research. But I know I just had lunch, so it's going to be a very light talk because I'm not going to talk too much in depth about what we are doing in the research. But I want to give you a brief overview of what we are doing, but in relation to what needs to be done in order to have a translational research approach in a sustainable way. So first of all, what is translation research? Everybody is aware of that, or कोई बताएगा? नींद खोलो, कोई बताएगा? Translation research. What do you understand by the word translation research? So anything out of research which you can translate, that means which you can apply. So there are various maybe I can read from here only.
Pointer. This one is a pointer. Chain भी होगा. Pointer.
Okay. So what is translational research? So when we say translational research, as I said in a very simple way, whatever research we are doing or whatever knowledge we have generated from a research work, if we can apply that, that is simply translation of a research. That can be taken in various, depending upon which field you are working on. So I have just listed some of the definitions which I found. So it is applying findings from basic science to enhance human health and well-being, because we are working in a biomedical research. So it's basically for the human health and well-being. So in other words, if you, I think most of you are aware of this terminology, bench to bedside. Suna hoga na something? Bench to bedside research. So bench means doing research and you take it to the bedside, means you are taking it for the use of human diseases. So, but what I think is that translation, the last one, the translation of promising results from clinical studies to day-to-day medical, because we are working in a medical, we are doing some clinical research. So we want to take it to the medical practice, and that is what translation is about. So now, how do we do translational research? So there are two approaches I'm just describing. So this will help you. So I'm not going to, as I said, I'm not going to bore you with what we are doing. But actually, if you take up any problem, as I said, to make your research more sustainable. Sustainable means you want it to have a long lasting effect. Then you need to select a good question or good research problem on which you should work. So from today's talk, hopefully what you will be able to get is how to plan your research and what kind of topic you should be taking up so that your research is sustainable. It shouldn't just die down after a few years. It should go on and on. So that is my main purpose of giving this talk over here. So bedside to bench approach means to address a clinical question. So I'm talking about biomedical research. That is why I have changed the terminology from just research to biomedical research. So whatever clinical issue, if you can address any clinical issues or if you can address any clinical need, then that means you are working on something which is needed in the society, not just for your own curiosity. It is important to address your curiosity as well. But as Dr. Prem already said, that we are in India, we are not from very like Western society, so we have limited funds. So we should focus on something which is also required in the society rather than just focusing on your own curiosity. Second is bench to bedside approach. So bench to bedside approach means whatever knowledge you have gathered, if you can just directly use it for human disease and management. So I'm giving you a few examples based on what we are doing at SGPGI. So as I said, how to frame a good clinical research question so that your research would be meaningful and could be sustainable also. So we are working on kidney health and maternal health. So now I'll give you example from kidney disease. Kidney disease is actually one of the, if you are aware, it is a huge global disease causing global disease burden, and it has many challenges, especially in the patients who are diabetic. Every one diabetic individual may be having this disease, but they may not be aware of it because in initial phase it is very asymptomatic, and by the time they know it, it is already too much late, and then reversing anything would be difficult than preventing something. So that is why it is very important to work on kidney disease, and it is not only causing a disease burden, but it is also a quality of life for the patient gets very, it's because they have to undergo the transplantation and all those things. So we have also done some research to establish that the existing parameters which we use to diagnose kidney disease are not enough. And as you all, I'm not sure if, because आजकल तो वो lab pathology कौन Dr. Lal's pathology and all, and people are without consulting doctors, they are even going for their own routine checkups, right? So you all know that for kidney, अगर मैं kidney health assessment कराना है, so just get the creatinine done or get the protein is coming out of the urine or not. These are the simple tests which are actually in routine use in clinics as well as in society. But there are various drawbacks of it, because whatever test we are using or whatever test doctors are using to diagnose kidney disease, they are not specific, and also they are not an early indicator. ये सब तब आते हैं जब already disease एक तरीके से established हो चुकी होती है. So we need something which can predict kidney disease or which can actually early diagnose it rather than. So as I said, the existing diagnostic methods are not enough, not specific. So how can we do that? So we I have received, I have been sanctioned a center of excellence from ICMR to just translate something and to accelerate the translational research program for prediction and diagnosis and therapeutics of kidney disease. So this is something which ICMR has given us in 2018, and there are 3-4 projects under this major objectives. So one of the aim of our center is to find out if we can early diagnose kidney disease in the patients who are at risk. So as I said, diabetes is a major risk factor for kidney disease. So this is the first aim. Achha, I haven't given this, sorry. So and then the other, so there are 2 objectives of this center. First, those who are at risk but have not developed kidney disease. So if we can diagnose early, we can predict in those patients, then enormous burden can be eased, because that way we are preventing kidney disease rather than curing kidney disease. So that is the first, that is early disease in high risk population. So early disease or in the population who are at risk of kidney disease. And then the subjects who already have kidney disease, there are problems in that too. It is not only the diagnosis which is a big problem, but what happens is that kidney disease happens in diabetic individuals, but it is not always due to diabetes. And that is very difficult to know unless you do the kidney biopsies. So that is a bigger challenge in diabetic individuals, because if you do biopsy in diabetic patients, अगर किसी ने सुना होगा कि अगर diabetic बंदा है उसको चोट भी लग जाती है तो wound healing is a big problem, big issue. So that is why diabetes जो है biopsy एक already invasive procedure है, and in diabetic individual it is even a bigger problem. So that means we need to do something that without biopsy, if we can diagnose what kind of kidney disease they have. So we have in our center two major aims: one, early diagnosis, and specific diagnosis without the need of kidney biopsy. So we have developed a non-invasive diagnostic method, which I am going to just show you in brief. So as I said, to get because whatever marker we are using right now, they are not directly, we are not studying kidney tissue, because we are studying something which comes out in the blood or which comes out in the urine at present time. But whatever comes out in the urine and whatever comes out in the blood, it can be coming from any tissue type, not necessarily from kidney. Are you getting my point? ठीक है? So जो blood में आप देख रहे हो कोई molecule, for example creatinine we are seeing in the blood, or creatinine is a bad example in this case, but any molecule which we are seeing in a blood, because when we say non-invasive, we want to avoid biopsy, तो हम क्या करते हैं, blood ले लेते हैं, उसमें check कर लेते हैं. पर blood तो सारे tissues से connected है, so it can be coming out from any tissue type, but we have to make something which is coming only from the kidney tissue. तो उसके लिए हम क्या करते हैं, we do kidney biopsy, ठीक है? And as I said, biopsy is invasive, so this is a real challenge. Biopsy करेंगे, we can diagnose, but doing biopsy is invasive, right? So how to evaluate without putting individuals at risk? तो अब डायबिटिक बंदों को हम रिस्क पे नहीं डालना चाहते हैं, but we want to evaluate their kidney tissue. That means we are talking about non-invasive. So this gives us a good clinical issue, clinical question. So मेरा बताने का सिर्फ इतना मतलब है कि when you are going for a research, try to find out this type of clinical needs which are unmet, which we need to resolve. Then your research may have a good impact. Also it will be meaningful. So this is how you design your research question. तो इसको कैसे किया for students? We have to do a lot of literature search, but besides literature search, you have to involve community or the stakeholders, not community, stakeholders. कौन है वो? Doctors. So जब आप डॉक्टर्स के पर्सपेक्टिव से चीज़ों को देखते हैं, उनसे पूछते हैं, then you will be in a better state to find out a good clinical question. So this is for the students who like to pursue research, how you frame a good research question for your sustainable translational research. Second question, maternal health. Maternal health के लिए भी खास तौर से जो women's who have already gone through this phase, उनको तो मालूम ही होगा. Most of other people must also know कि pregnancy में क्या होता है. Pregnant women कहते हैं ना कि दूसरी जिंदगी मिल गई, जो बच्चा हो गया, कहते हैं ना. तो उसका कारण क्या होता है? Because there are various diseases in pregnancies जो कि आपको पहले नहीं होती हैं, but at the time of pregnancy you start getting, like preeclampsia, eclampsia is a सबसे worst disease होता है, क्योंकि उसमें बच्चे की भी जान को खतरा होता है. Unless you just तुरंत डिलीवरी नहीं करते हैं, मदर को भी खतरा होता है, ठीक है? So preeclampsia is a huge issue in pregnancy besides gestational diabetes, और छोटे-छोटे बहुत सारी चीजें होती हैं that you can only know जब woman pregnant है, and that too clinical symptom आता है तब आपको पता चलता है, that by that time you can only wait and watch कि हाँ भाई ये बढ़े नहीं ऐसे. लेकिन अगर आपको पहले से पता चल जाएगा तो लाइफस्टाइल इंटरवेंशन से आप बता सकते हैं, because every person has a different need. Personalized medicine का अप्रोच है. So it is very important to know कि प्लेसेंटा यू नो ऑल, आई थिंक एवरीबडी इज अवेयर ऑफ प्लेसेंटा, सो आई डोंट हैव टू एक्सप्लेन व्हाट इज प्लेसेंटा. सो प्लेसेंटल हेल्थ का अगर हमें टाइम टू टाइम थ्रू आउट द प्रेगनेंसी इफ वी कैन समहाउ स्टडी प्लेसेंटा पूरे है उसमें, तो कब क्या, क्योंकि हर समय चेंज होता है प्रेगनेंसी में, क्या होता है एवरी मोमेंट थिंग्स आर चेंजिंग इन प्लेसेंटा, प्लेसेंटल डेवलपमेंट में. सो इफ यू कैन स्टडी प्लेसेंटा मे बी एवरी ट्राइमेस्टर, हमको एक अच्छा नॉलेज हो जाएगा कि कहीं कुछ रिस्क आ तो नहीं रहा है, बिकॉज इट इज समथिंग विच इज रिलेटेड टू प्रेगनेंसी, इट इज नॉट देयर इन द वुमेन बिफोर प्रेगनेंसी. ठीक है, बट प्लेसेंटा की बायोप्सी ना कोई करेगा ना कोई करने देगा. इट इज एन एथिकल इशू, इट इज बेसिकली रिस्क फॉर वुमेन, क्योंकि प्लेसेंटा को आप नहीं देख सकते. सो अगेन बाय नोइंग द प्लेसेंटल एडॉप्टेशन we can easily know कि क्या हो रहा है प्रेगनेंसी में, but because of the limited material availability we cannot study placenta during pregnancy. Pregnancy हो जाती तो placenta तो मिल जाता है, लेकिन during we cannot study. So again comes a need of a non-invasive source of placental tissue is required, right? So I have given you two clinical questions: non-invasive way to study kidney tissue and non-invasive way to study placenta without biopsy. So this is the challenge we took कि हम इसको करते हैं, because it has a huge societal and patient के perspective से it is a good research question. So then we come up, so that means we are talking about liquid biopsy, ठीक है, without actually dissecting or taking out a part of the tissue. You are doing a liquid biopsy in circulating जो biofluids होते हैं, that is या तो blood और या urine samples. कैसे करेंगे? एज आई सेड कि ब्लड एंड यूरिन में तो हर तरफ से चीजें आ रही, नॉट इन यूरिन, यूरिन में तो सिर्फ यूरिनोजेनिटल ट्रैक्ट का आ रहा है, बट ब्लड में तो इट इज कमिंग फ्रॉम एवरीवेयर. सो हाउ कैन वी नो कि वट एवर वी आर स्टडिंग इन ब्लड इज फ्रॉम प्लेसेंटा और इट इज फ्रॉम किडनी टिश्यू? सो फॉर दैट वी फोकस्ड ऑन एक्स्ट्रा सेल्युलर वेसिकल्स और एक्सोसोम्स, आई शुड से. Extracellular vesicle का exosome is a subtype of extracellular vesicles. So extracellular vesicles, yeah, these are what I am talking about are the nano vesicles which are secreted from by the every cell of the body. Cells की जितनी भी body में जितनी भी cells होती हैं, they secrete exosomes in the blood and in urine samples. ठीक है? But again, it is secreted by everything. तो कैसे हम करेंगे? So this is I am just going to show you. So without going into the detail what are exosomes, but this is just to show you how exosomes are released. So this is a cell, and as you can see these red dots, red vesicles, these are exosomes. So these are multi-vesicular bodies are formed, that is multi-vesicular body here, and then it fuses with the plasma membrane and it comes out in the circulation. So that is how cells के अंदर बनता है and बाहर आके release होता है. ठीक है, एंड वाई इट इज इम्पॉर्टेंट? सो दीज आर सम ऑफ द प्रॉपर्टीज ऑफ एक्सोसोम्स. तो एक्सोसोम जो सेल निकालती है उसका एक तरीके से यू कैन से इट इज अ मिरर इमेज ऑफ दैट पर्टिकुलर सेल्स. ठीक है, तो इट कंटेन्स बायोमॉलिक्यूल्स ऑफ द ओरिजिनेटिंग सेल्स. सो समहाउ इफ वी कैन कैप्चर द एक्सोसोम्स व्हिच आर कमिंग फ्रॉम प्लेसेंटा एंड कैप्चर द एक्सोसोम्स व्हिच इज कमिंग फ्रॉम किडनी टिश्यू, तो हमारी प्रॉब्लम सॉल्व हो जाएगी. ठीक है, बट दीज आर सम ऑफ द प्रॉपर्टीज. So contains biomolecules of originating cells. So they have microRNA, they have mRNA, and there were some debates, but now it is proven that they have DNA too. They have some protein too. So these biomolecules, which are of the tissue origin, that can be studied in exosomes, which are coming in the blood. It protects the, and because there are, if you want to study some microRNA, RNA in urine, urine is a toxic environment. ठीक है, तो सब डिग्रेड हो जाएगा. बट इफ दे आर इन दैट वेसिकल, सो दे दैट डोंट गेट डिग्रेडेड. सो दैट इज अगेन अ ब्यूटी ऑफ एक्सोसोम्स. Exosomes protect the envelope functional components from degradation and transfer. So another important quality of exosomes, I want you to understand and to know these three important properties of exosomes, which makes them not only a good source of biomarker but also therapeutic option. Why? Because ये exosomes क्या करते हैं? Now you can imagine there is a vesicle which has various microRNA or mRNA in it. These vesicles comes out from one particular tissue or one particular cell, and then they go and they are taken up by the recipient cells. तो जब recipient cells में वो microRNA या mRNA ट्रांसफर होता है तो क्या होता है? have you read transcriptome, translation तो मालूम होगा सबको, transcription मालूम होगा सबको, I hope. So क्या होता है कि when these microRNAs also regulate gene transcription. ठीक है, everybody is aware of that. MicroRNA are small 20 nucleotide sequence. They can regulate gene transcription. So when microRNA from one cell is transferred to the other cell, तो उस सेल का भी ट्रांसक्रिप्शनल चेंज हो जाता है, दैट मीन्स ट्रांसक्रिप्टोम चेंज हो सकता है. सो दैट मीन्स बाय ट्रांसफरिंग मॉलिक्यूल फ्रॉम वन सेल टू अनदर, सेल टू सेल कम्युनिकेशन का रोल प्ले करता है एक्सोसोम्स. सो दीज आर द ब्यूटी और गुड क्वालिटीज ऑफ एक्सोसोम्स व्हिच मेक्स देम अ वेरी पोटेंशियल target as a diagnostic tool and as well as a therapeutic tool. So how to proceed? So now we have a good clinical question. We want to proceed. So I'm just giving you example of maternal health, what we have done. As I said, exosomes are released from the entire body type and they are in the blood circulation. Not only in the blood circulation but in all the biofluids, including milk, saliva, everywhere they are present. So what we have done, so similarly placenta also releases exosomes in the blood. Then we used some specific antibodies which are only present in the placental tissue and we capture these in the blood samples itself. So placenta, so this is placental alkaline phosphatase. Alkaline phosphatase is present in liver and you are aware of that. So similarly there is a placental specific alkaline phosphatase also. So we use this antibody to selectively capture exosomes. Immunoprecipitation, everybody is aware of? Malum hai? Good. So similarly, on the similar concept, what we have done is we have used these antibodies or blood may suppose you have various type of exosome but only those which express this specific placental derived exosomes or placental derived genes, they are captured. So what we have done, we have taken up and then this here you can see we have characterized these exosomes. By nanotracking analysis, we can know the size of vesicles which we have enriched. These are electron microscopy images and these are some of the markers we have done. So just point of showing this slide is that we have done the whole transcriptome of these exosomes. So this is a heat map. I'm not sure if you are aware of it. When you do microarray or when you do NGS, next generation sequencing, so then instead of looking at one gene at a time, you can check the entire whatever expressed over there in NGS, and in array also you have a defined set of, because this array what we have used have lot many microRNA, you can study them in one go. So we have captured these placental derived exosomes and then by using this array technology, we have profiled their microRNA and also mRNA using NGS. So this is the whole transcriptome we have studied. And what we have done is we have captured them in first trimester of the same woman, then second trimester and third trimester. So what we know from that is with increase in trimester how this transcriptome changes. And transcriptome generally tells you about the phenotype also, that how it can be related to the phenotype of the cell or the placenta. So how the gene expression changes with time during pregnancy. So what we have done here is normal pregnant woman, first trimester, second and third trimester. So we know the profile of a normal pregnant woman कि कैसे होना चाहिए, कैसे gene change होनी चाहिए in pregnancy. Now suppose अगर हमें नहीं मालूम है and we take any woman and we check this placental derived exosomes at 3 time point, if it deviates from this profile, तब फिर एक alarm raise होगा कि हाँ, there must be something different than what it should be in the normal case. So this is one way to look at it. Similarly, we have done protein analysis. I'm not going into the detail, but very briefly what we have done is, so when, as I said, you have taken up placental derived exosome with the blood. For kidney, urine is the best source, but you cannot check the entire urine content for some reasons because urine कैसा होता है, is a toxic environment. So whatever rare proteins, rare molecules you have, there are chances that it get degraded. So in order to protect that from degradation, what you can do is you can study the exosomes which are present in the urine. Okay, so that is what we have done. So this I think I can skip, but what I want to emphasize here is that in the urine we have checked, we have found a protein after proteomic profiling. We found a protein WT1, which is Wilms tumor protein. It is specifically expressed in the podocyte cells of the kidney, and we found that this WT1 positive patients, or the patients in which WT1 comes out to be positive in exosomes, they have deranged kidney function. Now, so this slide is very interesting, and it tells, I just want to tell you how you can predict a patient, diabetic patient, कि उसको kidney disease होगी कि नहीं होगी. That is why this slide is very important. So when we were doing our study, what we found is that some of the patients, if you see this black bar, some of the patients have this protein expressing, so they are diabetic patients. Some of them have proteinuria, means protein in the urine. They don't have, negative means they don't have protein in the urine. So there are three groups of patients we have studied. पहले patient में diabetes भी है, kidney disease भी है, as you can see here. Second group में diabetes तो है, but there is no protein. That means kidney is normally functioning. Third case में there is no diabetes or kidney disease. So तीन group of patients को हमने study किया, and what we found is that कि जिसमें diabetes भी है, kidney disease भी है, all of these patients have WT1 in their exosomes. पर वो population जिसमें diabetes तो है लेकिन kidney disease अभी तक नहीं है, उसमें 50% have, 50% don't have. So इसका मतलब क्या हुआ कि जिसमें WT1 है, they are at risk, they might be at risk of kidney disease, जबकि अभी है नहीं. और जिसमें नहीं express हो रहा है, they may be better off, उनको भी chances नहीं है next 3 to 5 years. So to prove that, what we have done, we have followed up these patients. तो इन patients को हमने करीब 2 years के लिए follow up किया, and we found कि WT1 is an early predictor or early prediction of developing proteinuria in subject with diabetes. So as we have predicted that those who have WT1, they develop kidney disease earlier than those who do not have. So exosomes is telling you something ahead of time. So that is how you can prevent. So if you know कि इनको होने की संभावना है, you can tell that patient and they can take precaution. So we have published couple of papers कि how exosomes can be useful for in renal injury or kidney injury prediction. Another example for kidney health. So ये सबको मालूम है what is this? Can anyone tell me, students? Physiology का कुछ तो होता होगा, kidney nephron. ठीक है. So this is just to show you कि this is the one entire. This is अरे वाह, very good. Proximal tubule, and you can see this one nephron. So each and every epithelial cell lining which is towards the lumen, they secrete exosomes in the urine, and that is how we can capture. अगर हमें किसी particular cell type का exosome चाहिए, we can use particular specific protein expressing in that region, and we can sort out these exosomes. So that is what you can do, and this is the mechanism. I don't want to go now. The clinical question. So again, so this is just to tell you how you take an approach. So clinical question to bench. Now we have established in that exosomes contain markers which can predict kidney injury, right? But there are some limitations. We cannot do that in human. We cannot check everything in human. So we have to come to animal model. So in order to prove that hypothesis, that kidney injury can be predicted, we have created an animal model. Diabetic, this is streptozotocin-induced diabetes in animals, and then we have followed them for few weeks till they develop kidney disease. So we have collected urine samples at 3rd week, 6th week, 9th week. So at 9th week they started developing, showing the signs of kidney injury, not before that. Then we wanted to see whether the predictors express at week 3. अगर predictors express हो रहे हैं week 3 and week 6 में, then only there is a use. अब if the predictors are expressing in 9th week, there is no use, क्योंकि 9th week में तो हमें urine में already मिलने लगते हैं features of kidney disease. ठीक है? So we have done. So in order to ask this question कि whether urinary exosome microRNA analysis could be an early non-invasive indicator of renal disease, we have done this proteomic profile, NGS based array, where we have done before and after diabetes induction, and we found certain microRNAs which are promising, which can detect kidney disease. So here is an example of microRNA 451 which we found in exosomes at 6th week. So at 6th week of diabetes there is no sign of kidney injury, but this marker was up. And later on we found that it is related with kidney injury parameter also at 9th week. So this also again suggests that rise, every time we see a rise in this microRNA, that mice or that rat is going to develop kidney disease. So it is an indicator of kidney injury in animals. And we did some other experiments where we have shown that cell-protective role. So now in animal model we know that 451 predicted kidney injury in rats. But what happens in human? So now taking our knowledge which we generated in the bench to bedside. So this is how you take up. So animal model में हमने कर लिया. अब now we wanted to see whether this is true in humans or not. So we have isolated exosomes from urine, human urine, and we have validated using exosome specific markers. Then we wanted to check the expression of 451 in human urinary exosomes and whether it is associated with kidney disease or not. So in humans what we have done is we have just taken early kidney injury patients who were basically asymptomatic but some signs which indicates that they may be developing, because we needed a positive sort of control. So healthy control and kidney disease, but it is an early kidney disease, and as you can see there is a huge difference in 451 fold expression, and it is also related to GFR. GFR is glomerular filtration rate, which is an indicator of kidney disease. So basically microRNA 451 is associated with kidney disease also in humans, as we have found in animal model. And then we have bifurcated, because as I said there are two types of kidney disease, one which is because of diabetes and one which is without diabetes. So we have both type of patients which have no diabetes but kidney disease, and which have diabetes and also kidney disease. And in both these groups as compared to control, this 451 was increased. So whatever we have found in animals, we could see that yes, 451 has some relation to kidney injury. We are doing some studies where we are following patients who do not have kidney injury to just prove as it is as an indicator. Now, as I said that there are some limitations in human studies, we cannot do everything in humans. So, as I have shown you about WT1, which we have done in human studies just a few slides back. Now we want to see whether WT1 can predict kidney injury or not. This is a protein, 451 is a microRNA. So early kidney disease injury in rat was associated with rice. So we have again used the same animal model. So at baseline 6 week and this 9th week. So till here baseline and 6 week they do not have any kidney injury, but at 9th week they develop kidney injury. But if you see that urinary WT1 at 9th week it is increased, but not at 6 week data haven't shown, the 6 week it is not increased. So that is as opposed to 451 which we have found as a predictor, WT1 was only a diagnostic marker, not a predictive marker of kidney injury. So that is what we have done as compared to in rat model. So because in humans it is very difficult to follow up the patients. ये एक limitations होता है human study का. Although there are now large studies which are going on which can nicely follow patients, but while working on them I have realized it is challenging because some patients they drop out, they don't want to give their samples at a particular time. So you have to rely on human model, on animal models as well. So we have done some animal studies and then we have proven in rat studies. Now this is the first approach that we wanted to develop a biopsy, liquid biopsy for kidney and maternal, which we have done. Now bench to bedside. So whatever data we have gathered, we have basically translated it to bedside, and this is a big cross-sectional study which we have recently done in our center, and so it is a population-based study. So this is from we have taken two population because it is always good to check the effect in two different cohorts. So one of the cohort is from the Lucknow area, another cohort is from the Pondicherry area. So we have collaborators in Pondicherry, and WT1 story we have followed here also, and we have shown that WT1 strongly associated with high ACR. ACR is albumin to creatinine ratio, which is an indicator of kidney disease. So compared to urine, and these are NGAL and KIM1, you may not be aware, these are the tubular injury markers which are, people are studying more about it, कि ये हो सकते हैं. But whatever we have found in animal models, we have done in big population study, which is a cross-sectional study, where we found that WT1 strongly associated. WT1 can be a good diagnostic marker for early kidney disease. So some of these patients, they do not have any kidney injury to begin with, but we have found that they have high level of WT1, which later on they develop. So that means what I am trying to tell you here is that WT1 can diagnose kidney injury even the existing जो हमारे markers हैं, for example GFR या creatinine, so which are in routine use, they are normal in those patients. It is the WT1 which can early diagnose these patients. So this is again because this is from the exosomes which we have done. Second, this is actually one thing, the clinical use. The second use is the therapeutic options. So as I said, the exosome has a major role in cell-to-cell communication. It takes up molecule from one cell type and it transfers it to the another cell type, and that is how it has a potential to change the phenotype of that cell. So that means if you want to use if some cell has a disease phenotype and you want to convert it into a normal or a healthy cell, what you can do is you can inject these exosomes with that potential and then you can do that. So similar studies we have done in animal models. So as I said, 451 in our study we have found it as a protective role. So here, so this is actually we have done directly 451, and what we have found is that when we injected microRNA 451 containing exosomes, then these the parameters or indices of kidney injury were attenuated. So 451 enriched exosomes. So we have engineered exosomes and we have added 451 to it, and then we have found that this can attenuate your kidney injury. And this is a very recent paper where we have shown that you don't need to make a synthetic molecule. You can only you can just enrich exosomes from the urine sample and inject it in animals, and it shows us a therapeutic potential. So this is a pure biomolecule. It has no, we have not made it artificially. We have just enriched it with the urine exosomes of certain kind of patients, and we have injected it back in the animal models, and we have found that it has a therapeutic benefit. So this is actually, we have filed a patent for that, and then we have published this data. So this is again therapeutic role, exosomes secreted by stem cells. They attenuated diabetes in mice by changing the phenotype of the pancreatic cells. So these are just some of the slides which I wanted to show you the application of exosomes in diagnostics and therapeutic potential, and how you can frame your research questions so that it can be more sustainable. But there is more to it. So why we want to make our translational research sustainable? Because this is the whole theme of the conference. So sustainability is required not only to ensure long-term viability but environmental sustainability. And I think किसी ने तो बोला होगा इसके बारे में yesterday. I'm not sure if somebody has given a talk on environmental sustainability. नहीं हुआ. अच्छा. So basically translational research जब हम करते हैं, so we need to think about what sustainable approach we should use to make it long-term viable. So for that I have already shown you, you can take up some questions which are needed in the society or which are needed by medical practitioners, so that you can do that study and it has some meaning to it if it didn't fail then, right? And second is the environmental sustainability is very important, because this is actually in terms of translational translational research, this is less studied topic, but I think we need to give more emphasis to it, because the amount of water, consumables and plastic wares we are using, and nobody is paying attention to it. कितना ज़्यादा हम लोग single use plastic का इस्तेमाल करते हैं. So I think these things should be taken care of, because so that our research do not have a bad impact on the environment. So this is also very important. So how we can do it? We can, as I said, the biomedical research including wet lab, especially like wet lab I'm talking about, and clinical research has particular impact on the society, on the environment, and that can be minimized if we minimize the use of single-use plastic, water consumption, production, because it is about those labs which are heavily engaged in research. They should pay attention to it, and there are some drives now. It is called, I think, Green Leaf Drive. I'm not sure about the name, but they are helping this kind of lab so that you can minimize the carbon footprint because of your translational research. Now, as I said, to make your research more viable or long-term viability, it is very important to engage the community. When we just think about anything which excites us and we do research in our own lab, we may not be thinking about it, but we should engage local community by doing some, like as I said, we have done some population studies where we went into the population, we try to find out what are their needs and what are the problems associated with their community, and we try to work upon that. And similarly, you can engage stakeholders like medical practitioners, what we are doing, to find out their clinical unmet needs, which you can take up. So this is how you can engage the community to make viable. And then this is just few points which everyone should be aware of when they are planning for any study. Like it should have ethical practices, you should use ethical practices, community engagement, education, outreach, means whatever you are generating, it is important to have, you disseminate that knowledge through some workshops or something, so that more and more people are aware of it. They can cite it, not just to increase your citation, but so that more and more people can take up that research, and then whatever you have generated, somebody can take up to the next level and do it. And that is how जो भी आपने खर्च किया है या जो भी आपने carbon footprint generate किया है, that will be used rather than should die out. Open access collaboration, regulatory compliances, resource optimization, and continuous evaluation. So these are some of the key elements. So I wanted to just focus on these key elements rather than what I have presented before, because as I said, most of the students here are young students, and I think they are planning to take PhD as an option or research for their career. So बाकी तो चीजें आ जाती हैं with training and everything. But I think this is the most neglected part in research. और अभी तो नहीं, जैसे हम ग्लोबल वार्मिंग के लिए रो रहे हैं, थोड़े दिनों बाद we will realize the importance. But it is actually sad to see how much carbon footprint we are generating, but we are not paying attention to anything. We are only focusing on our research कि हाँ भाई data generate हो चाहिए, nobody is thinking about it. But I am sure चीजें आएंगी सामने जब एक भयानक रूप ले लेगी. तो before that, I just want to raise awareness among the students कि wherever you go, just keep these things in mind. Even if your guide don't tell you, you should keep these things in mind. कि whenever you do research, you should at least think about the environment and think about its sustainability. और because हम पैसा तो यूज करते हैं पब्लिक का, आप लोगों का पैसा होता है, so it should be better utilized rather than anything else. ठीक है. With that, I like to end. So these are some students who have passed out and some are pursuing PhD, who have actually generated data which I have shown over here, and some collaborators, current lab members and post PhDs, and some funding which I have received from various funding agencies. Thank you for listening. Any questions?
Any questions from the audience? Please go ahead.
mentioned about using exosomes and biomolecules, like not etheric molecules, biomolecules from urine of humans, and then using them in animal models. Don't they generate any immune response if we are not modifying them?
No, no, no. Actually, they are non-immunogenic, and that is why. Sorry,
it's working.
So they are non-immunogenic, and that is why they are actually undergoing in various clinical trials. You can find the various types of exosomes, especially stem cell derived exosomes, and those things are in clinical trials as well. So immunogenic response is very less. So they are not. That is what your question is, no? So it is. That is actually that is why people are using it, not only the biomolecule, the originally derived exosomes, but they are taking it and they are engineering it also, like introducing new biomolecule inside the vesicle. So as a drug vehicle also they are being used. It is carrier, nano carrier I should say. So अभी क्या होता है कि if you have seen various labs, because I am also collaborating with BITS Pilani, so they are preparing nanovesicles as a drug molecule carrier molecule. But what they have found is when they synthetically generate, there are some issues with it. So they have requested us to give them those stem cell derived exosomes so that they can put their molecule of interest in that vesicle, and then they are delivering it to their animal models. So these are less immunogenic. That is what, in short, the answer is that. Oh, that's a great side.
No.